Glaucoma relates to a group of conditions characterized by damage to the optic nerve and the retinal ganglion cells. High intraocular pressure accounts as the main significant risk factor. Another factor is related to similarities between glaucoma and Alzheimer's disease. Amyloid beta, the main component of the plaque found in the brains of Alzheimer's patients, has been found to coagulate and affect retinal ganglion cells. Amyloid beta peptides, generated by amyloid precursor proteins, are released as monomers into extracellular space and then aggregated spontaneously into oligomers, protofibrils, fibrils, and eventually deposit as plaque. Studies show that the most toxic amyloid beta form are not monomers or fibrils, but its intermediate. Identifying the number of damaging forms of amyloid beta and reducing their effect on retinal neurons will help close the gap in knowledge on how critical the amyloid beta elicited damage is. The purpose of the study was to characterize different forms of amyloid beta in a human retina of an angle closure glaucoma patient. Retinal sections around the optic nerve were treated with primary antibodies of the human amyloid beta that recognizes the 114 related to the N-terminus of the amyloid beta peptide that identifies toxic amyloid beta intermediates. Total human amyloid beta 142 to identify the whole peptide. We treated the tissue with brilliant blue G marker to stain monomers and theophylline tape to stain amyloid beta, fibrils, and plaques. Results show a subtle accumulation of amyloid beta monomers on the retinal pigment epithelium, the choroid, and some retinal regions. Amyloid beta intermediates were found in all retinal layers and the choroid. They appear to surround the cell and accumulate in sheet extracellularly, mostly near the retinal ganglion cells. There was minimal coagulation of amyloid beta intermediates with monomers. Coagulation between amyloid beta 142 and theophylline T was observed in the choroid, outer nuclear layer, inner nuclear layer. There was a substantial accumulation of total amyloid beta 142 in the retinal pigment epithelium, the choroid, the nerve fiber layer, the outer nuclear layer, the inner nuclear layer, and the inner layer membranes. RMNSA confirms the presence of damaging amyloid beta intermediates within all layers of the retina in glaucoma.